Microbial diversity can be defined as the range of different kinds of unicellular organisms, bacteria, archaea, protists, and fungi. Various different microbes thrive throughout the biosphere, defining the limits of life and creating conditions conducive for the survival and evolution of other living beings. The different kinds of microbes are distinguished by their different characteristics of cellular metabolism, physiology, and morphology, by their various ecological distributions and activities, and by their distinct genomic structure, expression, and evolution. The diversity of microbes presently living on Earth is known to be high and is thought to be enormous, but the true extent of microbial diversity is largely unknown. New molecular tools are now permitting the diversity of microbes to be explored rapidly and their evolutionary relationship and history to be defined. The purpose of this article is to define the scope and highlight major themes in current understanding of microbial life and to describe recent progress in expanding knowledge of the evolution and biological significance of this organism. Microbial diversity considered the vast array of microorganisms, the smallest forms of life, which exist everywhere. The three primary groups of microorganisms are bacteria, archaea, and eukaryotes. Bacteria and archaea are prokaryotes with their genetic material held in a single chromosome. In eukaryote, most of the genome is held in multiple chromosomes. Over 11,000 species of bacteria have been identified using microscopic identification of cell shape and metabolic activity, gram staining techniques, and genetic identification of RNA and DNA sequences. There are 500 named species of archaea divided into two phyla, the Eukaryote and the Crenarchaeota. There are 8 supergroupings of eukaryotes. All of them include single-celled organisms, and 5 are entirely microbial. Microorganisms are actually composed of every different and taxonomically diverse group of communities: archaea, bacteria, fungi, and viruses. The members of these groups or taxa are distinct in terms of their morphology, physiology, and phylogeny and fall into both prokaryotic and eukaryotic domains. They constitute a broad group of life systems inhabiting the known ecosystems on Earth, terrestrial and marine, including geographical location considered to be extreme or inimical to life. The latter comprise for such areas as habitats with high salinity, alkalinity, acidity, high and low temperatures, high pressure, and high radiation. Considering the adaptability of microorganisms to grow and survive under varied physico-chemical conditions and their contribution in maintaining the balance in ecosystem, it is pertinent to catalog their diversity as it exists. The inability to visualize them with the naked eye precludes effective classification. As such, using the available tools, microorganisms are broadly classified into prokaryotes and eukaryotes and subsequently into various taxonomical units depending on the resources available and required. The sustenance of life on Earth depends on maintaining the diversity of microorganisms. Human intervention is resulting in depletion of biodiversity, and many hotspots are also fast losing their endemic biodiversity. While specific data is hard to come by, it is likely what loss of macro life forms also results in loss of the associated microbial species, symbiotes, as well as the rhizosphere colonizing microbes. The significant contribution made by microorganisms in ecosystem sustainability as well as the industrially important biomolecules obtained from them, antibiotics, anti-cancer drugs, enzymes, biofuel, and various other compounds, implies that cataloging them is imperative. However, a simple and effective microbial identification system is still far off. The available tools for classification and identification of microorganisms rely on a number of different technologies. This chapter provides an overview of taxonomy tools for understanding prokaryotic and eukaryotic microbial diversity. The diversity of microbial communities varies within habitats as much as between habitats. This variation can even occur within a few millimeters, suggesting that microbial diversity encompasses more than the documented evidence available. Hence, biogeography is gaining importance as a field of study from microbial diversity point of interest. Main reasons have been postulated to explain this phenomenon. Due to the innately small size of the microorganisms, environmental complexity plays a major role in determining diversity. Spatial heterogeneity is like to lead the formation of mini niches within a habitat. Recent tools like metagenomics aid in biogeography studies by providing information on nucleic acid sequence data, thereby directly identifying microorganisms. Therefore, the phylogenetic information can be used to compare microbial diversity profile across habitats. Generally, diversity within a particular location and in a community is called alpha diversity. Beta diversity measures the community composition between two or more locations, while gamma diversity applies to a region across continents and biomes and is larger in size than that used for measuring alpha diversity. The evolutionary relationship of microorganisms is called phylogeny. Understanding phylogenetic profiles of microbes become a daunting task because of their small size and the lack of particular indicators that could serve as markers. Some proteins and genes are considered as evolutionary chronometers, which measure the evolutionary change. Currently, the 16S rDNA sequence is considered the most reliable for measuring evolutionary relationships in bacteria and archaea, and the 18S sequence for fungi. However, it is necessary to choose the correct protein or gene for such studies, such as a gene or protein should have certain features which make it most appropriate or deriving evolutionary relationship. The most important criterion is that it should be present in all members of the target group and be functionally homologous in the organism. The molecule must contain regions of conserved sequences for comparison purposes. The changes in sequence data must be at a slow enough rate to permit measurement so that it may also reflect evolutionary change for the entire group. The universal phylogenetic tree based on the comparative ribosomal RNA sequences. The phylogenetic tree representing all living organisms shows that evolution of current forms of life occurred from a common ancestor, the universal ancestor, depicted by the root. Two domains are of prokaryotic systems of life, the archaea and eubacteria, in contrast to previous systems of classification wherein the prokaryotes were confined to a single kingdom. However, it is intriguing to note that genomic studies have shown the archaea to contain unique gene sequences which are not present in bacteria or eukaryotes. Certain genes are also shared between all the three domains. The genes required for core cellular functions are called the ones which are necessarily for survival of a cell and could have arisen from the common ancestor. The divergence of the organisms represents the differences in genetic sequences which could have become fixed in each group as they evolve. It is also postulated that earlier, each GT played a key role in transfer of genes between organisms early in the evolutionary history. Our new understanding of microbial diversity will allow us to cure diseases, engineer and conserve our environment, manufacture better products, grow more food, colonize other worlds, and so much more.